फैन क्यों बंद कर दिया? फैन क्यों बंद कर दिया? फैन क्यों बंद कर दिया?
Good morning, everyone, and a warm welcome to all of you in this workshop. On behalf of Flavinlabs Pvt Limited, I am delighted to welcome you all to this 3 days online workshop on mushroom spawn production. This event is designed to be your first step towards professional mushroom cultivation, offering insights into the fundamentals of spawn production, hands-on techniques, and innovative approaches to maximize success in mushroom farming. Flavinlabs is a technology-driven research and development company recognized by Startup India and certified with ISO 9001 in 2015. I'll kindly request you all to please register your name in the comment sections of the YouTube and continue with us. In this workshop, we'll go through fundamentals of mushroom spawn and cultivation techniques. Step by step, we'll go through the guidance on the spawn preparation with the live demonstrations, which will be demonstrated by our instructors. And then we'll study about the effective methods to identify what should be the method to control the contamination, because mushrooms are really susceptible to contamination, and they require really uh, protection for contamination. Then we'll go through the insights into business opportunities and strategies for scaling mushroom cultivation. The time duration for this workshop will be for 1.5 to 2 hours. Next, we'll go through, in the next slide, there is the introduction, and I'll introduce you to our Flavinlabs. Flavinlab is a technology-driven research and development company recognized by Startup India and certified with ISO 9001 in 2015. We are dedicated to translating cutting-edge laboratory research into accessible, real-world solutions while empowering communities through training and entrepreneurship. Um, Our mission is to enable participants, from students to aspiring entrepreneurs, to gain practical knowledge and industry-oriented skills in this growing sector. Over the next few days, you'll learn step-by-step methods of spawn preparation through the live demonstration, which will be demonstrated by our instructors, and discover effective strategies for contamination control and explore business opportunities within mushroom cultivation. This workshop also features interactive lectures, video-guided experiments, and comprehensive study materials. By the end, you'll receive a globally recognized certification, adding valuable credibility to your portfolio. In the next slide, we see that what our Flavinlabs provides. What are the services we give? What else we provide? At Flavinlabs, we focus on cutting-edge research and development while also delivering innovative products and services in the field of science and technology. The services which we provide are academical and industrial oriented. The first in the academics is summer winter training, which is for 15 to 60 days, or which is for 1 to 2 days also. There is workshops like this one. The workshops will be for 1 to 2 days, or this one is for 3 days. So it depends on what are we doing in the workshop. There are certain training and project programs which we offer, which is for 3 months to 6 months. Then there is dissertation and thesis support. You can also come to us for the help. We provide help for your dissertation and your project and thesis support, thesis submission. Then the last service is the biotech finishing school program, in which we provide students with all the requirements which a newbie or a fresher requires to come into the field of working other than academics. When you have to join some industry or when you have to go in the field of job and work, so we provide that finishing touch to your academics and to your uh, portfolio. So this is all about biotech finishing school program, which is which in which we also provide a guaranteed job after the program is finished. Then we come to the industry oriented. There is food and feed testing, in which we do tests for the food and feed related. Then is the spawn production. Spawn production and spawn sale. We also produce and also sell the spawn. Spawn is basically the seed that contain the living uh, fungal mushroom mycelium that is required for the growth of mushroom. So we'll go through all in this 3 days workshop. You'll learn all about it, what is spawn and what is another other things. Then comes the mushroom cultivation. Yes, we also provide mushroom cultivation in Flavinlabs. Then comes the mushroom farming consultancy. If someone is going in the field of startup, if they want to go in the field of mushroom cultivation, so what are the things they need, what are the things they require to succeed in this field, is what we do here. We consult them, we um, make them learn what are the things they used to have in mind for mushroom farming. Then comes the tissue culture banana plant, for the banana plant. Then here is the training modules: microbiology, molecular biology, biotechnology, mixed modules. These are the modules which we provide for microbiology, for molecular biology, for biotechnology, mixed module, nanotechnology, mushroom cultivation, cordyceps mushroom cultivation, biochemistry, statistical analysis for life sciences, linkage map and QTL identification, herbal product formulation. These all are the training modules which we provide in the Flavin Labs. The duration for these modules can be are very flexible according to you, whatever you are in certain with. Short terms are 1 to 2 days, then 3 days, then 7 days, and 15 days, whatever you prefer. It's up to you. And then long terms are for 1 month, for 45 days, for 3 months, for 6 months. And these are the training modules and time duration which the Flavin Labs provides to the students. Then comes the training mode. The training modes are both. We go for the both modes, offline and online. For the offline, the location is near Pandit Deen Dayal Upadhyay Nagar, Sultanpur Road, Arjunganj, near Ikana Stadium, Lucknow. And the online mode is through the links that we provide. The online modes are enhanced with the experimental videos, the PowerPoint presentations that we give, the study materials, and the live lectures that we provide you. What will you receive after the online or offline training modes? What will be given are the hard and soft copies of the certificate. Which will you ask? Whichever you ask, either the hard copy or the soft copy. Then we'll be providing you the personalized letter of recommendation. If you request us, we'll provide you that also. And the letter of recommendation is highly valuable in your portfolio. It is very valuable if it is coming from such a lab which is renowned by uh, mm, ISO, which is certified by ISO and BIRAC and BIONES BHU and all these MSME. So it is very valuable for you. And then comes the welcoming kit. If you are coming online and want to learn offline, sorry, if you are coming offline and want to learn offline and want to go for the offline mode, you will be given the training kits, which will be having uh, lab coats and all. Will provide you everything in the training kit. Certificate details and workshop participation. The participation of workshop is free of cost. Anyone can participate in it. There is no cost for it. Then comes the certification. For the certification, there are two kinds of charges that we offer. Uh, One is the digital certificate charge, and the other one is the hard copy charge, hard certificate. So in the digital certificate, you'll be wanted we'll want to you to pay 299 rupees for the digital certificate. We'll provide you the digital copy, which will cover all the taxes and the processing fee for the certificate. Then comes the hard copy certificate, sorry, which will be of 699 rupees. And the hard copy certificate will be premium and laminated, and which will also include all the printing charges, all the lamination charges, and dispatching, and all those charges will be included in the 699 rupees. The certification of Flavinlabs or the workshop is valuable. And why is it so? It's because the lab will provide you a ISO certified laboratory and will provide you a credible certificate, which has a strong credibility. Then the certificate will be premium quality, so professionally designed and laminated. So you'll get the premium quality certificate. Then the certificate is very valuable for your portfolio. Makes your efforts count, because again it's a certified laboratory. So, and the certificate is also globally valid. You can apply it everywhere on your LinkedIn or wherever you want to apply it, and also in your job. Then here are the peoples which are in background working for you and instructing you for all about this workshop. Here is Dr. Sandeep Kumar, the director and R&D head of the Flavinlabs Pvt Limited. Then there is Kuldeep Mishra, senior research associate, workshop convener and trainer. He'll be guiding you through the workshop. Then comes Ms. Shivangi Suman. She'll be guiding you, she'll be instructing you about all the mushroom cultivation in this 3 days of workshop. Then I am here as Avishka Srivastav, your anchor for the workshop. Now, further going to start the workshop as for the mushroom cultivation. I'll be handing over the screen to Ms. Shivangi Suman for further explaining you the mushroom spawn production.
So, good morning, all of you. My name is Shivangi Suman, and today I welcome you all to this 3-day workshop on mushroom mushroom spawn production. So, without wasting any time further, as we have discussed and gone through the introduction, let's dive into the workshop. So, the main focus of the workshop is live demonstration. So, mainly we'll be dealing with the live demonstrations of things. We'll be demonstrating you how spawn is being produced, how spawn cultivation takes place. Along with the live demonstration, by the time live demonstration will be going on, we will also be giving you the theoretical knowledge about what is spawn. Firstly, we'll be discussing it about the mushroom and then the spawn. After that, you will get to know what are different substrates and why different substrates are used for different kinds of mushroom. So if if you are joining this workshop keeping the entrepreneurial thing in mind, that you want to set up your own spawn production plant, so we'll be dealing with that in detail one by one. So we'll be giving you everything that is needed for spawn production. So let's get into the presentation. So this is the basic module of that what we are going to discuss in this presentation. If we take the workshop schedule of 3 days, so the first goal of the workshop will be what is a substrate preparation. First, what is substrate, and then how substrate is being prepared for spawn production. The next goal of this is about the chemicals. Ki what other things instead or matlab, apart from substrate, what other chemicals or what other essential compounds that are mixed with substrate to enhance the growth of spawn, because alone substrate cannot provide all the nutrients that are needed by the spawn to grow. So there are other essential chemicals that are needed, and the next goal will be covering about those essential chemicals. The third thing that we'll be discussing about will be bagging. So how the bag is being prepared. Bagging is basically this substrate that we are using and mixed with all the other essential chemicals is now either filled in a polythene bag or is filled in jars, and that process is called bagging. So the next goal will be bagging. After the process of bagging, the next thing is sterilization. So as all of you know, if you are from the biology background, you understand what is on sterilization. And if you are not, then let me tell you, sterilization is the process that makes any environment contamination free. So we are sterilizing after bagging to make the substrate absolutely contamination free. Contamination free means any kind of biological contaminant that can spoil the spawn later on. We'll be sterilizing that. The next, after sterilization, the next step is inoculation. So we'll be dealing with inoculation after sterilization. Inoculation is done. Inoculation is basically we'll be adding uh, the mushroom into the substrate. We'll be adding that, and after that the mycelia will grow in that substrate and it will provide prepare spawn. Then after inoculation, the next step will be incubation. So incubation is basically we provide adequate amount of temperature and moisture for that mycelia to grow and to develop and to produce better spawn. Basically they will grow and they will develop, and then that thread like structure will be spread throughout, and then it will produce the spawn. And the final will be the business aspects. As we have said, the motto of this workshop is to help people who wants to set up entrepreneurial thing. So we want to help them set up those business aspects. So we'll be at the end we'll be dealing with the business aspects of those things also. So this is basically our workshop schedule. Now in this next half of the slide, I hope you could see there is a laboratory setup. So there are certain equipments, and now we need to discuss about these equipments as well. So the fourth goal is sterilization, and as you can see, this is this is autoclave. So autoclave is basically the machinery that is used for the sterilization process. After that, our fifth goal is inoculation. So we cannot inoculate any, fungus or bacteria, anything into open environment. We need a contamination free chamber for inoculation, for the process of inoculation. And for that we use laminar flow hood. So basically laminar flow hood, that space is absolutely contamination free, so any other microorganism else than the fungus, that is the mushroom that we are going to inoculate, except that no other fungus grows in it. We need to make that sure for the better spawn production, otherwise your spawn will be spoiled. So for that we need a laminar airflow. And the as we have discussed about the sixth goal, we need incubator. So this is incubator, and this is what this will provide adequate, there are different kinds of incubators. So for mushroom we need to maintain the temperature, as well as certain kinds, spawn we need temperature. If we are cultivating mushroom, then we need to maintain humidity as well as temperature. So here we are using kind of incubator that only maintains temperature. So there are different kind of incubators, one that only maintain temperature, other that maintains temperature as well as humidity. That's called humidity chamber. And there are different kinds of incubators that comes along with the shaker. So this is a basic kind of incubator, and this is used for setting up the temperature. So the adequate temperature is set up to maintain that thing, temperature thing, and then helps in growing the spawn. So this is our today's agenda, what we are going to discuss today. So first we'll be dealing with introduction of what is mushroom and then what is spawn. After that, since we have done with what is spawn, we'll start with the substrate preparation. So we'll be showing you the demo, live session of how the first step of substrate preparation takes place. With that, with meantime, we'll be discussing the theory things, all the other things, and after that we'll be discussing with the theory, and then at the end we'll take a case study, and by the time of 6:30 to 7 we'll be taking question answers. So if you have any kind of question along, by the session goes on and you want to ask something, so keep posting your question either in the comment section, or you can send your questions in our group. So we'll be answering you there. But it's better if you post your questions in the comment section. So by 6:30 to 7, we would like to answer those questions, and we'll deal with your queries. So let's get into the uh, actual workshop thing. So first we'll be discussing with what is mushroom. So first important thing that we need to understand about mushroom is a type of fungus. And we'll discuss what is fungus actually. So it's a type of fungus, and it basically reproduces through spores. And as we know, we go to restaurants and everywhere, even in our houses, mushroom as a food is very common now. So it's often used as either as food or as medicinal purposes of mushroom. I think most of us are aware of how mushroom is used as medicines. And if you're not, then let me tell you that mushroom is a very a booming source and a really good natural source of medicine as well. Along with this, there's a huge number of industrial applications of mushroom. Apart from food and medicine, there are other use of mushroom also in industries. So if we think about mushroom as food, why we take mushroom as food, why there are so many people and doctors and nutritionists are recommending that you should take a mushroom? Because first, very important, mushroom is very low in calories. So if you are taking 100 grams of fresh mushroom, you will get only 20 to 30 kilocalories. Along with this is a very good source of protein as well, and it provides really good kind of essential amino acids in the body. And for people who are vegetarian, source of protein, there are very few vegetarian things that has really good source of protein. You can eat a lot of different components of food. Maybe you can take pulse and other grains, and then you will get all those 2 to 4 grams of protein. But only if your intake is 100 grams of mushroom, then it will provide you with 2 to 4 grams of protein. Along with this, it has good amounts of micronutrients like vitamin B12, vitamin D, antioxidants, so that improves the immune system. It helps with people who are struggling with weight management, so it's good source that also helps in weight management. Along with this, it's very good source of food for people who are struggling with heart health, because this is really low in cholesterol. Along with this, it has high potassium content in it that helps in maintaining the blood pressure. So all these things make mushroom a really good source of food. Apart from this, if we discuss about medicine, then there is a medicine called cordyceps. There is a mushroom called cordyceps, and cordyceps is really good for the stamina of people, so it improves the stamina. And there are other medicinal properties of cordyceps as well. So there are different other kinds of mushroom also that has really high medicinal properties. And if we talk about industrial applications, so mushroom, when mushroom is being grown in industry, so there's a substrate on which mushroom grows, and that substrates really works as, it enhances the quality of soil if it is mixed with soil later on in farm fields. And also it works very good as compost. So that's a great industrial application of it. Along with this is used in waste management and in bioremediation. Bioremediation means, in the removal of pollutants and in removal of heavy metals or dyes also. So these are the industrial applications of mushroom. Now, as we have discussed, ki mushroom is a fungus, not a plant. So the basic difference between, plant and fungus is that plants are autotrophs. Autotrophs in the sense that they have photosynthesis, they have chlorophyll in them, and due to chlorophyll they perform photosynthesis. And by performing photosynthesis they make their own food. Now if we talk about mushroom, mushroom does not have chlorophyll, so they cannot make their own food. And since they cannot make their own food, they are relied on some other source for their food, to fulfill their nutritional things. So for that mushroom, since we discussed that plants are autotrophs and mushrooms are heterotrophs. That is, they feed themselves on dead and decaying matter. And how they feed themselves, we'll discuss that in detail later on. But the basic thing that we need to understand is that they decay the waste products. For example, if their substrate is wood, or their substrate is leaf, so they decay the leaf, they you retrieve nutrition from there, and then they grow. So basically, for the growth of mushroom, they need something dead and decaying kind of thing, what they will degrade, and they will take their nutrition from there. Now if we discuss about the parts of the mushroom, then there are certain things that are really important to be discussed. That the first top part, this where pilus is mentioned. So pilus is the cap like structure. This is umbrella like structure, and the main purpose of what is the main purpose of pilus? So it is used for protection of gills. So it's basically, it's fruiting body ornamental kind of things, and the main purpose of it is to protection of gills. So what are gills? Gills are, thin blade-like structures, and they are very compactly arranged by by side of each other. So, what is the main role of gills? Is that gills has a special kind of tissue, and that tissue is called hymenium. This tissue helps in the preparation of spores. So, the cap-like structure, the pilus, and the gills prepare the spores, and then this release the spores, and this cap-like structure, pilus, helps in the release of the spores. So basically, and what are spores, that will be discussed later. So this gill-like structure helps in the preparation of spores. So millions and billions of spores are produced once. In the lifetime of mushroom, they produce billions of spores. After that, this is the structure. What we are discussing about is stipe. Stipe is also stem or stalk. So the main purpose of stem or stalk is that it provides the support for cap. It helps in elevating the cap, so the cap does not fall down, and it also provide the elevation to the gills so that it can work properly and prepare the spores. The next thing is mycelium. So inside the stalk, the stipe, there are thin fine line structures. Then those are myceliums, and myceliums are made up of fine thread like structures. Those thread like structures are called hyphae. And these hyphae and mycelium all together is mainly responsible for nutrition absorption. So they absorb nutrition. That is how, as we have discussed, that is how the mushroom takes nutrition, and this is how mushroom sustain itself. So for sustenance and for maintaining of absorption of nutrition, they mycelium is needed. And now we discuss what are spores. As we have said, key gills prepare spores. So spores are the reproductive units of mushroom. And basically spores are the reproductive unit of fungi, but since we are discussing mushroom here, so we'll be saying right now key spores are the reproductive units of a mushroom. And these spores are very small, light microscopic structure, and they can easily float in water or in air. And since it's very low in weight, it's really floats easily. So these are the seeds. But unlike seeds, if we talk about seeds, so seeds have nutrition stored in it. So if we sow a seed in the soil, it grows because it has nutrition in it. But spores does not have any kind of food stored in it. So once it gets on surface of something, maybe surface of wood, then it will start growing its mycelium. That mycelium will degrade the surface of that and then extract the nutrition out of it. Basically, spore has the outer surface of the spore is hard, and it is hard so that it can sustain different environmental condition. And when it gets the right kind of environmental condition, temperature and moisture, it could grow itself into mycelia, and then later on that mycelia will grow into the fruiting body. So that is what spores are. So, as we have discussed, these are the basic parts of mushroom: cap, that is pilot; stem, that is called stipe or stalk; gills. Gills are the structures, fine blade-like structures that prepare and then release the spores; and the mycelium, that is responsible for nutrient absorption and the supports growth. Mycelia basically support growth. Now there are different kinds of mushroom. There are some kinds of mushroom that are edible. That, for example, edible kind of mushroom that we consume every day. For example, button mushroom, shiitake, or white oyster that we are going to discuss about today. So there are different kinds of mushroom. These are edible kinds of mushroom. There are mushrooms with medicinal properties, like cordyceps and turkey tail. And of course, along with this, there are certain kinds of mushroom that are very poisonous in nature. So poisonous mushrooms are like, they have hallucinating properties, or they have toxins. And even sometimes touching a mushroom or taking just a bite of mushroom can be really dangerous, and it can be fatal. Means it can lead to death also. So these are all different kinds of mushroom that are present. So before cultivating, or even if you are going to the wild and you want to pick a mushroom, it's really important to know about what you're picking, because there are so many kinds of mushroom in the wild that are absolutely fatal for human beings. Now, along with this, we say that mushroom play a role in nutrient recycling in ecosystem. This means that, of course, as we have discussed, that mushroom feed itself on dead and decaying matter. So for that sake, it decays. For example, if we are talking about wild, there are lots of leaves are fallen down in the autumn season, and if there is no fungus kind of thing, they will not be get degraded. So fungi basically feed itself on decaying all those dead and decaying matters. So it's also called recycler in the environment. So this is all about what is mushroom. So this is how we have introduced mushroom. Now it's a very important thing that we need to discuss, and that is what is mushroom spawn. So mushroom spawn is basically the seed material of the mushroom. But as we have discussed earlier, that spore is also, spores are the reproductive unit of mushroom. So what is the spawn and how this is different from spores? So spawn is basically the seed material of mushroom, and spawn needs a clean carrier to grow itself. So that clean carrier is most of the time some agricultural grain or other agricultural substrates that are used as the clean carrier, that are used as a substrate for mushroom to grow. So this spawn takes that sterilized green carrier and then it get fully colonized on that surface, forming the white thread-like structure, and that white thread-like structure is basically mycelium. And once we put this white thread-like structure in other substrate, like for example in sawdust or in compost, then this white thread-like structure, this mycelia, spreads into the eventually it produces the mushroom, that it grows into a fruiting body of the mushroom. And the substrate that is used for this process is basically needs to be sterilized. Why it needs to be sterilized? Because supposedly the component that we are using, any substrate that we are using, if it's not sterilized, then for sure it will have some kind of contamination of maybe some other fungus like trichoderma, which is very common, or some bacteria or other thing. So it will compete with that mushroom for growth, and eventually it will lead to it will suppress the growth of that specific fungus that we want to grow, and the other thing like bacteria, mold, or that trichoderma will grow eventually in that. For that we need to take a clean substrate. So any substrate that we are using for spawn production, that needs to be sterilized. And that what is the purpose of using spawn? Because a good spawn can actually lead to a very good growth of fruiting body. If your spawn is not of high quality or is contaminated, then eventually it will hamper your fruiting body, your mushroom production. So basically, what is spawn? Spawn is the seed. Now, how it is different from spores? So spores is good if you want to take on some real high research project or in breeding, but it's very slow and it's really unreliable. And if you want to go for commercial production of anything, for commercialization, spore is not an relevant option, because you will be on uh, you know tired on things ki okay it will grow, but you're not sure that it how it will come out. Because in wild the things are very different. We are not focusing on strategic growth of anything, but when we take this on commercial growth, it needs to be strategic, and sports cannot be a reliable source for strategic growth of mushroom. So keeping that as an alternative, what in spite of sports we have grown spawns. So spawns are the seedlings that are basically used for fast and predictable growth of mushroom. So what is it? It basically skips the early stage thing. For example, if you're taking spores, so spores will go and then it will form mycelium, then mycelium will grow and develop itself, and after that it takes more and more time to grow the fruiting body. But since spores has already mycelia wrapped around the substrate, the growth of fruiting body becomes really easy and really fast. So why we are using spores in the place of, I mean, why we are using spawn in the place of spores? Because this is more commercial, more easy, and more tactable in keeping in mind the commercial things of growth. Now how will you know that what your spawn is of good quality? So if you are doing mushroom production, sorry, spawn production, and if you have to make sure that the quality of spawn that you're growing is really good, so if we think of the morphological things about keeping in mind, so the most important morphological things are the appearance. You need to see how is the appearance of your spawn. After that, how it is smelling. Smell is a very important thing. And then the texture of your substrate, because you cannot make sure about the texture of the spawn after that mycelium is grown. So you will have to know about the texture of the grain that you're using. So the first thing that you'll keep in mind is about the appearance. So your spawn needs to be complete white, even, and cottony. So the mycelia is cotton like structure, so it needs to be cottony and ropey, covering the whole of almost the grain that you're using as substrate. It needs to cover the whole of the grain. So the appearance needs to be absolutely white, cottony white, and even. And the substrate needs to be fully covered. That shows a good quality of spawn. It's an indicator that your spawn is of good quality. If you're seeing that there is some green or black patches, or there is pink patches, or there is on your white color, I mean the spawn is white, but there is some wet or slimy kind of patches. If you could see, it means that your spawn is started getting contaminated. So discard that specific bag of your spawn, because otherwise it will affect all the other bags that you have kept in your room. So first, keep in mind about the appearance. And the most important indicator, I mean, if it is getting green or black or pink patches, it's easy to recognize. But if it is getting slimy or wet, it's sometimes really difficult. So there needs to be someone who is constantly checking, is it getting slimy, or is there some kind of bacterial growth that we can see. Because since most of the time those bacterial growths are also white, so it becomes really difficult to differentiate. So that needs to be kept in mind if you are going for spawn production. The next thing that is very important is smell. So the room in which you have kept your spawns for growth, the important thing is that when you enter the room, you get, of course there will be different kind of smell, but the smell needs to be fresh and mushroomy, and it needs to be earthy kind of smell. If you are entering the room, or if you're getting inside one of your stand and you can smell something sweet or sharp, or there is some ammonia kind of smell or alcoholic smell, it means there is some kind of contamination. So this is very important. And so the it means there is some contamination, and you need to check again that. uh, and if we are talking about the texture, then the texture of the grain, we, as we have said, ki appearance, smell and texture matters. So here texture of the grain matters. So the substrate that you're using, it needs to be separate and shakeable. If you're using a kind of substrate that is compactly packed together, or making a brick kind of structure together, it sticks together, it's going to be a bad indicator, and eventually it will lead to spoilage of your spawn. So this is very important. These are the important things that you need to keep in mind. So basically, these are the things that need to be kept in mind if you are going for the spawn production. Now, the one thing that we have mentioned in at the bottom, that no light and no air is needed for spawn production. Here it means that mycelia does not need any kind of light for their growth, but it does not mean that it needs no kind of air. Of course, it needs oxygen, but it needs filtered kind of oxygen. So as you can see at the top, we have stick some filter kind of thing. So it will let only filtered air to pass through it, because if contaminated air will pass through your jar or through your bag, it will lead to contamination and spoil your mushroom spawn. So this is a thing that needs to be kept in mind, that you don't need a light, but you need air, but filtered air for spawn production. Now, before moving to the next theoretical slide, we need to start getting about what is, we need to get into the substrate, how we are going to prepare the substrate. As we have said, the first goal of ours is substrate production. So we will be getting into substrate production. So in that, we will show you that how we are taking wheat as our substrate, and wheat is the most commonly used substrate also. So in the next slides, we'll discuss that why wheat is taken, and why we take for different mushroom different substrates are needed. So we'll start we're taking the substrate. So now we'll show you, demonstration will start. So in that demonstration process, we'll show you how you need to take, in what quantity you need to take the wheat, how you need to boil it, any kind of substrate, any grain, how you take, how you add water to it, how you boil to it, how will you know that your substrate is boiled now, and now you can add different kinds of chemicals to it, and later go on with your other things. So let's dive into the practical aspect of it. And for practical aspect, we will be dealing with all those things like let's start get into first thing, that is substrate selection. As we have discussed, we have selected the substrate as wheat. We will discuss later on why we have selected wheat. So let's get into the thing, and let's discuss that how boiling things go on. So keep be up in the workshop and learn most of it, because this is basically planned keeping in mind, if you are planning to set up your spawn production plan, even if you're a student, it's going to be really helpful for you. because we are dealing with the theoretical portions and all the scientific things that are needed to be understood about it. But it's really good for entrepreneurial thing as well. So it's going to be helpful for both. Stick with it, learn, and if you want to take the certificate, you can, the link is shared in the community. I hope all of you are in the community, so it is already shared in the community. So let's dive in the live aspects. So as you can see, we are going to weigh. So this is the wheat that we're using as substrate. So we have weigh 200 grams of wheat. And now we are going to wash it. So you need to wash the wheat really carefully and neatly. Though we are going to sterilize, of course, but it's really important that you wash it clearly, because there are we are taking it from the farm, so of course it has all the contaminations like dust and everything, and it's very common in the wheat farm that we have trichoderma also. Of course we'll sterilize it, but apart from that we need to wash it really carefully and nicely. As you can see, we are washing it with multiple times with water. It cannot be done that you have washed it once and it's over. No, you have to wash it until it's completely clean and it's dirt free and dust free. So as you have seen, it's completely cleaned. We have washed it properly. Now we will, keep the, we'll transfer it into some container and we'll boil it now. So, for 200 gram of wheat, we are adding another 300 ml of water, and we will let it boil. So it will take around 30 minutes or so for it to be boiled. And I hope you will be able to see continuously that how it getting boiled and how it get, at what amount and at what texture it needs to get converted so that it can be used as a substrate for spawn. So you will see it. Because you could see, you'll be able to see it's getting boiled. And so basically mushroom, spawn production or mushroom production is a time taking thing and it's a tedious thing. People who are absolutely lazy or who does not want to give time, constructive time to something, it's really difficult for them to do spawn production or mushroom production. So you need to be, basically very much scheduled and do everything on proper time for this process to happen success, for the success of this process. Now I hope you could see that the water has started getting boiled. Water is boiling, and of course within 20 to 30 minutes you will be able to see how the uh, wheat has changed its texture and as now is boiled. You will see within 20 to 30 minutes that uh, by the time the it is being boiled. Let's continue with the theoretical portion of mushroom spawn cultivation production. So if we discuss that what is basically mushroom spawn substrate. So what is substrate? Substrate is basically both food and home for the mycelia. So how we say it's a food and home for the mycelium is basically it provides two essential things to the mycelium for its growth and for its development. First is nutrition, because since we know mycelia has no nutrition on its own, it need nutritions to grow and to develop. So the substrate provide nutrition, and the next thing that the substrate provide is it provides surface area, because suppose it does not have any. Sorry for the disruption. There was some little commotion was there. So let's again start with. So what is actually spawn substrate? So basically, spawn substrate is basically both food and home for the fungus, that is mycelia. So how it, the two important things that we need for the mycelia to grow is for the nutritions and the surface area. And substrate provide both of these, the nutrition also and the surface area. As you could see in the other video that is being shared, that now the wheat has started changing its texture. So the nutrition that we are discussing about is the chemical energy that mycelia needs to grow. It needs some nutritional source like carbohydrate, protein, and other things for growth. So that is provided by the substrate. And the next thing that needs its surface area. So it needs some space for mycelia to replicate itself and to grow. So all these things are provided by the substrate. So there are different kinds of substrates that are being used. So either we are using general grains, cereal grains as substrate, or we are using other agricultural byproducts. So either we use cereal grains or we use agricultural byproducts. So what are cereal grains? Cereal grains, for example, either we use wheat or we use rice, other kind of different kinds of millets. Brown rice is also used. Different kinds of maize and millets are used. So how cereals are good for that? Because cereals are rich in carbohydrate, protein, minerals. So they provide, they act as a mini nutrient packet. Along with this, their size is small and uniform. So that small and uniform size gives a good kind of contact surface point for mycelia to grow and replicate itself. And along with this is easy to kept in a jar and to be sterilized. So that is the reason that we use cereal grains. There are different other kinds of substrates that are also used. These are agricultural byproducts. So these byproducts are maybe, you can use corn cobs, or you can use sawdust, you can use paddy straw. But paddy straw and sawdust, these are basically better for mushroom cultivation and not for spawn production. Because first, it's very difficult to sterilize them, and sterilization is a very important technique, for spawn production. Along with this, this has uh, more complex kind of structure, and this is not uniform. And uniformity is very important for spawn to mycelia to grow around it. Now you can see that the wheat is getting boiled and it's changing its texture now. So all the people who have any kind of confusion or they want to ask some kind of question, they can post their questions into comment section, and we will be taking that by 6:30 to 7 a.m. PM. So please post your questions. We are looking forward for it. So these, what are the advantages of using agricultural byproducts? Is basically they're cheap, they're abundant, and they're sustainable. But limitation is that they are very hard to sterilize, and they are not uniform. That leads to ununiform growth of mycelia, and that leads to not really good growth of fruiting bodies later on when we use this as a spawn, seed. So why grain is more popular? Because it is easy to sterilize. It gets, it's, you can wash it easily. And when you autoclave it, so the heat penetrates through the grain, and then it gets sterilized easily. So sterilization and cleaning is easy. Again, it is very much nutrient rich, so it supports healthy rich mycelia, because mycelia head does not have anything of its own. And again, even spreading is a very important thing in spawn production, and this even spreading is absolutely supported by this small uniform size. Again, grains are also widely available, and they provide really good high surface area. So if surface area is high, mycelia growth is high. So this is vice versa, relatable to each other, and that is why most of the time grains are chosen as substrate. But along with grains, why we are choosing grains, there are other factors also which decide why we are choosing certain specific kind of strain for certain specific kind of mushrooms. For example, We are talking about button mushroom. Button mushroom is very widely available. People generally eat what kind of mushroom we are consuming nowadays. All of us consume is basically button mushroom in India. So button mushroom, prefers organic matter, decomposed organic matter. For example, manures and straw. So you would have heard that for mushroom production, people are using compost kind of thing, that are manures, or they're using straw, rice straw or wheat straw. These kind of things are used basically for button mushroom. Why they're used? Because in the wild, button mushroom prefers aggregus bisporus. That is the scientific name of button mushroom. So it prefers all those nutrient rich, composed, decomposed organic things. So when we are growing that in the lab also, we try to imitate what kind of nutrition it consumes in the wild. So keeping that in mind, for mushroom growth we use manure and straw kind of things. And for spawn production of button mushroom, either wheat is used or rye is used, because that provides same kind of nutritional composition to the mycelia also to grow. And either wheat or rye provide very good kind of colonization for button mushroom. Similarly, if we talking about oyster mushroom, so the natural habitat of oyster mushroom is decaying wood, decaying wood, straw or agricultural waste. So if it grows in the wild, it will grow on things like somewhere that where wood is being decayed, or there is some kind of straw or agricultural waste. So what kind of spawn substrate it prefers? It prefers either wheat or millet grains. So it grows very good on wheat or millet grains. Similarly, if we talk about shiitake prefers hard logs in the sense that hard woody parts of the plant. So for shiitake, the better kind of substrate that is preferred is sawdust. So before, at the time we are choosing what kind of substrate we should use for specific kind of mushroom spawn, we think about the thing, or we study about the things, or it's generalized SOPs are created that how it used to be grown in the wild, and same kind of things we try to replicate in the laboratories as well. Now there's a figure that you could see. This figure actually explains that how mycelia absorbs nutrition. So this is a figure that shows ki how mycelia absorbs, uh, or any kind of fungus, how it absorbs nutrition. So it's a very important thing that we need to understand. As we have discussed also, that mushroom thrives on dead and decaying things. So how it thrives on dead and decaying things, and also we discussed that it's called recycler. So what does this recycler mean? So basically this figure is being discussed here and it's given here. So it says ki hold on for a moment please. Yeah, so we were discussing that how mycelia observes anything. So how it gets its absorption. So basically mycelia does not have any kind of mouth or stomach. What it is, it's a thread like structure. But this thread like structure is mycelia is what? It's a group of hyphae, and hyphae is thread like structure. So these hyphae release certain kinds of enzyme that break down starch or cellulose or protein into simpler sugars or amino acids. So this process of breaking down of starch or sugar, starch into sugar or proteins into amino acids, is basically called external digestion. And when the surface is externally digested, the the hyphae gets inside it, and through the hyphae walls, mycelia absorbs nutrition from it. So, and by absorbing that nutrition continuously, mycelia grow itself and form the whole substrate colony, that it gets completely, uh, colonized throughout the substrate. So this is how mycelia absolutely gain its nutrition. Now the thing that needs to be discussed here is grain spawn is most widely used. uh, why we use grain spawn? Because it is easy to sterilize, as we have said, it is nutrient rich, and it ensures in uniform distribution of mycelium. So these are the important points that make grain a good substrate for mushroom production. I mean spawn, mushroom spawn production. Now we are using wheat as a spawn substrate, as we have seen that in laboratory also, as we are boiling it, you can see in your on your screen that this wheat is being boiled. So as we are So wheat is being boiled. As you can see, it will, change its texture by the time. So it will, we are using this wheat as substrate. So why we are using wheat as substrate? Because, first and foremost, wheat is the most worldwide used spawn substrate. And why it is? Because first, it provides balanced nutrition. Its nutritional profile is very much balanced for any kind of mushroom production. So, I mean, apart from wild, when we are doing commercial cultivation, so most of the time for spawn substrate, wheat is preferred. First reason is its nutrition profile, and second reason is it's really easy to handle. Because if you are dealing with something commercially, you need to make it simple. If it's very complex and lethargic and something that is very, tedious to deal with, automatically it will cut down your profit. Because if it's very much tedious, you need to engage more and more people to it. Anyway, mushroom cultivation or spawn production is something that needs you to be timed and scheduled. And again, if it's something you're using as substrate, for example as corn cob, so it's really difficult to first sterilize, and then to, the mycelia to grow along with it, and even the mycelia grows with it, its bagging is difficult. So all these things, and after bagging also, when you're, using this spawns as seed for mushroom cultivation later on, this, because it's very chunky and big in size, of course it is going to hamper the later on, production of mushroom also. So what we need, your seed needs to be very much easygoing and easy to handle. And of course, along with this, it needs to have really high nutrient profile. So if we, discuss the composition of wheat and its role in mycelia growth, the first thing first that is very important to deal with is that wheat is has 60 to 70% of starch. Carbohydrate content is really high in wheat, and carbohydrate is very important for mycelia growth. So this 60 to 70%, profiling of carbohydrate in wheat really supports growth of mycelium. And this is the main reason that wheat is used as the main source, as the most widely used source for substrate. So, these fungus, these mycelia produce certain enzymes like amylase, and it breaks down starch into simple sugars, and these simple sugars become easily available for the same mycelia to grow and flourish itself. Now, if we talk about protein, so protein, protein also supply amino acids for fungal enzymes and structural proteins. So enzymes are needed to, digest the substrate further. So once they have digested the carbohydrate, now they also suck protein from it, and now from that sucking the protein, they produce enzyme, and that enzyme helps in further degradation on the surface. So protein is also important, and it has significantly high amount of protein to 15%. After that, if we discuss about fiber, so fiber structure is not really, directly attached with something nutritional, but because it's a bulky kind of structure, so it provides that mycelia, you know enough of space to wrap around itself and to anchor itself on the fibrous structure. So so again, this fibrous structure is also very important. Where if we are talking about lipids, so it supports cell membrane synthesis, and that cell membrane synthesis is very important for, mycelia. So of course, the cell membrane will be synthesized of hyphae, and then again it will be able to take better nutrition from the substrate source. Now, if we are discussing about minerals, so there's magnesium, calcium, phosphorus, potassium, and these enzymes act as cofactors for spawn production. And as we are going to discuss later on in this, that we even add calcium and phosphorus additionally, because calcium and phosphorus are really needed for spawn production. Now you could see that our wheat has boiled really nice, and once it will get completely boiled, it will turn into some pearl like shiny structure. So once it gets nicely boiled, nicely cooked, you will be able to see some, now of course you could see this has swollen really beautifully, and it has got that shiny texture of pearl like. So this is how you will decide that your wheat has boiled completely. Or now, you need not to be, need not to overboil it, because if you will overboil it, it will get mushy and it will stick together. And as we have discussed, that your substrate needs to be shakeable, it needs not to stick together and form cakey kind of structure. So how you will know that it has been boiled completely as a substrate? So you will take one or two grains in your hand, and you will press it, and once you will press it, you will see that it actually does not get completely mashed, nor it is very hard. It's somewhere mushy, but not actually mushy, and it has got that shiny surface because it has absorbed water, and it has turned into pearl-like structure. So what we were discussing about is about the mineral things, that of course we even additionally are going to add some kind of chemicals into it, and those chemicals will be rich in calcium and phosphorus. So calcium and phosphorus is really needed for spawn production for any kind of mushroom. So even though if you change the substrate, the process is somewhat going to be same every time. You have to follow the same procedure, and same kind of processes and same kind of chemical mixing is done time to time for this thing, for spawn production. Now we will be discussing with key why wheat is used for mushroom spawn production. As we have discussed that we can use different kinds of things, but along with that nutrient profiling, what are the other reasons for that we use wheat as mushroom spawn production? First thing first, nutrient thing. So it is absolutely rich in nutrients. So it is really rich. It has good amount of carbohydrates, 60 to 70%, that really helps in making it a good source of nutrition for spawn production. Along with this provides a good surface area. So good surface area means it's not very much bulkier. It's not very small in size. So it's provide enough kind of space for your mycelia to grow and cover itself. And since it's a grain size, it's of perfect size and everything is perfect, it's you know more grains is absolutely equal to faster colonization. And now if we, what does faster colonization mean? So faster colonization means fast growth of mycelia, and fast growth of mycelia means that very fast mycelia will get spread around each of the grains. It will cover the grain, and now the spawn, all the grains that we have in one packet, one bag, is absolutely covered by the mycelia. So they have occupied all the space, and this needs to be processed very fast. So why fast colonization is important, that we are going to discuss. Because first, if your mycelia is growing fast, it's getting enough of nutrition and it's growing really fast, so it will be out competing the contamination. Suppose your microbes or your molds or your trichoderma, it grows with it. So there can be a chance that there is some kind of contamination that is left there. or your substrate has started degrading due to any reason. There can be any reason, there can be some faults, or there can be some mistakes that we have done unknowingly. So if your substrate is of right kind, and you have provided, it's not wet, it's not dry, it's somewhere in the middle, 50 to 55% of water content that is needed for spawn production, so it has maintaining that, and your substrate is good, and your mice, you have provided right kind of fresh mycelia, and it's absolutely growing fast, so it will outcompete all the trichoderma. Though trichoderma really spreads fast, and it's really difficult to compete trichoderma, but yet if it's growing fast, it may outcompete trichoderma, different kinds of molds, and it can absolutely outcompete different kinds of molds and microbes, yeast and bacteria. So it will spread, leaving no room for other nutrient invaders to grow. And if it has grown completely, so the chances of spoilage gets really reduced. If it takes really large amount of time for growing, colonization takes very much time, then the chances of contamination always increases. So fast colonization also leads to, first, it will outcompete the contamination. Second, and very important, fast colonization also leads to uniform mycelia spread. So if you want your mycelia to spread uniformly, it needs to be colonized fast. Slow colonization leads to patchy growth, and then uneven or reduced yield. Basically, spawn is going to produce mushroom. So if it's your colonization is fast, your mycelia is going to spread fast, and then your mushroom is going to appear even. If your colonization is slow, your mushroom is going to be patchy, and it's going eventually it is going to lead to uneven or reduced growth. Now you could see CERN is checking that how it has boiled properly or not. So in the meantime, when you keep your spawn for boiling, you need to keep checking it, that is it too mushy, or is it too hard, or its texture is fine, not really mushy, not really hard. You need to keep checking this time to time. And once it's boiled, as we have said, it gets shiny pearly kind of structure. Now let's come back to again. So why fast colonization is important. First, it will outcompete the contamination. Second, your yield will be consistent. Third, since your yield will be consistent, it will reduce the crop loss and cost, of course. Because if your one bag gets contaminated, the chances of contamination of other bags really increases. Along with this, if your spawn is is contaminated due to any reason, and you sold those spawns, so now the production of the mushroom is going to be affected. And spawn, of course, if a spawn is of bad quality, it will affect crop production a lot. So keeping these things in mind, it's very important to keep your substrate, choose your substrate wisely, and most of the time that substrate needs to be wheat. So as we discussed, why fast colonization important, we need to keep that in mind, why we need colonization of mycelia to be fast. If we take one live example, so generally oyster mushroom spawn colonized in bulk in 10 days if it is provided with wheat as a substrate. And generally the green molds that take 12 days to grow. So since your mycelia has colonized within 10 days, or by 10 days, by 12 days the chances of growing that green mold is now very slow, or very low, because since all the gaps are covered by mycelia, so the chance that green mold will grow because your wheat is boiled too much, or you have too much humidity, or any kind of 50 to 55% of humidity can also lead to it. So it will get absolutely reduced. So keeping that in mind, fast colonization is really important. Now the third reason sorry why we use mushroom mushroom wheat as mushroom spawn production. So it's easy to handle. Easy to handle means it's a grain size, so it's small, it's uniform, it's easy to mix with bulk substrates. So bulk substrates means, if when you are going to produce a mushroom, for mushroom cultivation you are going to use some kind of substrate. That substrate is going to be bulky. So if your if your spawn is of small size, it's going to be really easy to mix that small spawn, uniform size small spawn, to that bulky substrate. So it will get easily mixed, your seed will be provided to the substrate, and the growth of mushroom will be enhanced because of this. Now the next thing is availability. So as we know, you know, these things are really cheap. Wheat is really cheap and is widely available. This is also the reason that we use wheat as the mushroom spawn substrate for mushroom spawn production. So it's cost effective, it's widely available, and it's easier for even for farmers or for labs to retrieve it from anywhere, and it's very widely cultivated not only in India but all over the globe. So this is also the reason that it is used for spawn production. And again, the next reason is support fast colonization. So as we know, as we discussed in detail, that why fast colonization is really important. So this is also the reason that wheat is supported for spawn production. Now the wheat has boiled and it has got the texture of shiny white pearly kind of structure. So now we are going to filter this, and we'll spread this for getting dried. And after that, so the process of filtration is going to start. We'll see that now. I hope you could see it has swollen really beautifully, and it has got those pearly structure. I hope you understand what does pearly structure mean. So we have sterilized um the, bu- uh, the the container for filtering the wheat. It's very important that whatever you are using is absolutely sterilized. More than mushroom cultivation, spawn production needs sterilization. Oh, now see that it's not really mushy, that it's get completely crushed, nor it is very hard, and it has got those. It has really solid, that has absorbed water and got really beautiful, nice texture of pearl. Kind of pearl. Please keep posting of your questions if you have any question in the community. Either you post your questions in the group, or you can post your questions in comment section so we could answer it later on. We are going to take the question and answer session soon, so please do that.
You can send your questions to the group admin also.
So you could see we are filtering it. We'll try to remove as much as water we can remove. And after that we'll spread it on a filter paper. So basically filter paper will absorb all the remaining water. So we are evenly spreading it so that it gets dried. So we need not to dry it. Actually, we need to maintain the moisture of about 50 to 55% for the better mycelia growth. Because as we have discussed, if the wheats grains if it is sticky or it gets stick together, then it will lead to, if it has moisture content more than 50 to 55%, then it's going to be really wet, and it will hamper the growth of mycelia and will later lead to the chances of really high chances of contamination. So that is kept in mind. So it is even we have covered it with another filter paper, so any extra amount of water that we have will get absorbed by the filter paper, and we will keep it for overnight drying. You can also use 3 to 4 hours, but since this is a time which has a really high moisture outside, so we will prefer overnight drying process. So basically you have to see that in what time duration it gets about 50 to 55%. So 3 to 4 hours is also fine at places where humidity is really low. But since the place we are sitting has really high humidity right now because it's rainy season, so we prefer overnight heat drying of wheat substrate. So as you have seen, we kept it for drying. Now we will discuss that why we discussed why we use wheat. Now we'll discuss why we boil wheat before use. So the first thing first, that is very important to know, that boiling absorbs water into the grain. So for before boiling, the grain is really hard, and for any kind of growth, any kind of mycelia or something needs a bit of moisture for their growth. So boiling absorbs water into the grain, giving right moisture content that mycelia needs. So mycelia needs some kind of moisture for their growth and for their development and spreading, and that moisture content is nearly 50 to 55%. So for providing hydration, keeping the mind reason of hydration, we boil the wheat. Along with this, this softened grain coat. As we have discussed, that mycelia needs to penetrate into the substrate, and then it need to digest the substrate, and then need to take the nutrition from the substrate. So if the substrate is really hard, it's very difficult for mycelia to grow. So for that process, we boil the substrate, and so the covering of the substrate gets moisture and it gets softened, and it becomes easier for the mycelia to penetrate and to absorb the nutrition. The third thing, and the very important thing, that we need to understand, it increases its nutrient availability. So if the anything that is not cooked, for example, why we cook our food, we generally cook to enhance, make those nutrition easily available to us, because in the hard and raw form this is not going to be easily available to us. So to make this soluble and to easily available to us, we need to cook this. For the same reason, we are boiling the you know the substrate. So the heat will help into the partial breakdown of all the starch or any nutrient thing, maybe in protein, so that it will be broke down into amino acid, and then the starch, and for mycelia it will be easier to convert those starch into simpler sugars and to take the nutrition. So for making the nutrition available easily to the mycelia, this is also the reason that we boil any kind of substrate, basically wheat, as we are discussing with wheat right now, where it removes impurities. So boiling always helps in washing away dust, inhibitors, and excess carbohydrate that could cause contamination. Along with this, we know that any kind of agricultural product that we are dealing, there is very high chances of all kind of trichoderma kind of things and other kind of bacterial things and all that. Along with this, there is dust and all that. So spawn needs absolute sterilized environment. So for that also, for keeping in the impurities mind and in the in contamination in mind, we also boil it. So for keeping it, removing the impurities and the contamination, we also boil it. And to prepare for sterilization, so hydrated grains are easier to be sterilized. After this boiling thing and bagging thing, we are going to sterilize it in autoclave. So if it is not boiled, the grain is really hard, and the heat is not going to penetrate as easily as it is going to be penetrated in the boiled grain or any kind of boiled substrate. So for keeping that also in mind, we since it's hydrated well, its sterilization also becomes easier. So for and sterilization, as we have, removing the impurities is important, but sterilization is even more important. That is making it free from any microbial environment. So keeping all those things in mind, also we prepare. We boil the wheat. Now after boiling, once we have boiled the wheat, now what we need to do after boiling this? So once you have boiled anything, there are chances that it will become sticky or it will form lumps. So for that we need to mix different kind of chemicals. As we have said, we have the second goal is mixing of chemicals and other substrate. So the that goal is that since the grain has gone soft, so there are chances that it will clump together. Or after clumping, maybe if there are three or four grains that has come together and stick together, so the oxygen reach among them will be really reduced, and that will inhibit the growth of mycelia. So we make sure, even after boiling, that is why we have, as we have said, that we make sure that it has not boiled really into a mushy kind of thing. It needs to be boiled in a proportion that it's not really hard, but it's not even really soft. It has got that shiny, pearly structure. So so to avoid it getting lumping, we need to add certain kind of chemicals to it. Along with this, if it has high moisture content, it is going to be a breeding ground for different kind of bacteria and molds and fungi, and that will cause what contamination, that will lead to contamination for mushroom and for spawn. Along with this, we also need to right now when anything is boiled, during sterilization and colonization, grains release organic acids, and thus organic acids make the pH unstable, and this makes the medium too acidic. I mean, once you have sterilized and after that colonization is taking place. So during those colonization process, what we said, ki mushroom, the mycelia is going to rupture the upper surface of the substrate, and then of course if it is going to rupture the upper surface of the grain, it is going to produce some kind of organic acids, and these organic acids is going to make the medium really acidic. And once the medium gets acidic, it will hamper and slows down the growth of mycelium. So to avoid all these things, after boiling, we need to add certain chemicals, and those chemicals are calcium sulfate and calcium carbonate. Now today we are not going to add those chemicals because we are keeping it for overnight drying, so that the moisture content of the substrate reaches to 50 to 55%. Tomorrow when you will come back, in the workshop. In the next session, we will be adding certain chemicals to that boiled substrate, that is wheat. So there are two kinds of chemicals that is going to be added into it. So first, if we discuss key in what quantity we add, so for we weigh the wet grain, the grain that we have already boiled, we are going to weigh that. So if we talk about calcium sulfate, so 2 to 3% of calcium sulfate is added of the wet grain. So if the weight of the wet grain is 200 grams, so we are going to add 2.5% of that in calcium sulfate to it. Similarly, we are going to add 0.2% of calcium carbonate to it. So why we add calcium sulfate or calcium carbonate, that's also a thing. So first, calcium carbonate, if we discuss about calcium carbonate, so calcium carbonate basically acts as a buffer, as we have discussed that, the mycelium, the pH get unstable as the colonization starts. So the mycelia release the organic acids, and that organic acids makes the pH really acidic, and this acidic pH hampers the further growth of mycelia. So to maintaining that, we we add buffer kind of things to maintain the pH. So we need to maintain, mycelia needs alkaline pH 6.5 to 7, and calcium carbonate provides that pH 6.5 to 7 is that alkaline pH is provided by calcium carbonate. Along with this, this acidic, built up is prevented by calcium carbonate. So the calcium carbonate basically provides the stable environment for the mycelia to grow for the long term colonization, so that the whole of mycelia grows onto the surface of the wheat or any substrate. So for maintaining the stable environment for growth of mycelia, calcium carbonate is used. So that the pH is slightly basic, that is 6.5 to 7, and that too much acidity is neutralized. And if we talk about gypsum, so gypsum basically helps in keeping the grains free flowing. Keeping the grains free flowing means since they have boiled, so there are chances that they will stick together, with due period of time. So we add adequate amount of gypsum to it, so that there is enough of friction kind of thing in between them, so that they do not stick together and that make clumps. Along with this, gypsum also provide calcium and sulfur. As we said, we need additional nutrients also. Apart from the substrate, we need to add some additional nutrient also into it. So we add calcium, we add gypsum, and gypsum provide additional calcium and sulfur. Gypsum also helps in maintaining the pH stable. So two of these chemicals are added into it to make this look really better. Now, as we have discussed, we have boiled, and now this slide, of course, we are going to you will see we'll be dealing tomorrow. So now we'll be taking the Q&A session. All the questions that you have put up in the comment section, we are going to take that. So we are going to take a break of 2 minutes so that we could see all the questions, and after that we'll start the Q&A session. So be with us, bear with us for 5 minutes, and then we'll start with the Q&A. Now let's get into the Q&A. So let's take the first question.
The first question is it possible to grow cordyceps mushroom at beginner level with low budget?
Yeah, it's absolutely possible. The quality can be a problem because cordyceps has some composition called cordycepine, and at the start, the composition and the quantity of cordycepine varies in different kind of cordyceps. If you want to set up a basic cordyceps, as a beginner, you just need an incubator with a light and laminar. Basic these are the basic things that are needed, so you can do the setup. And export market is really good for cordyceps, so you can do that.
Second question from the participant is how much area required to set up a spawn production?
So for spawn production, you need a 10 by 10 room, incubator and laminar. So this much is enough for spawn production. You can start your spawn production in small scale from there, and I am sure it is going to give a good profit.
Third question says for medicinal mushroom production, what is the scope?
It's really high for medicinal, mushroom production. There are even better market for export of it. And there is dwindling market for in India for medicinal scope, but it's the scope of mushroom market is really high, and medicinal aspects is really high in export, so you can do medicinal mushroom cultivation for export purposes. Keeping that in mind, it's going to be good.
One of the participants is asking that he or she is staying outskirts in the outskirts of Mumbai. Is what is the commercial scope there in the outskirts?
I am sure it's going to be high, but I think apart from starting, mushroom cultivation, you should start with spawn production. Why? Because, the chances of spoilage of mushroom is really high. It gets spoiled within 4 to 7 days, and it's tough to market in 4 to 7 days because I'm sure the market of Mumbai would be completely crowded of it. But, in all over India, there is a good gap of spawn production. So the good quality spawn is, I think, is going to be a good market. You can do market study, and I'm sure spawn production is going to be a good thing for that.
One of the students is asking that I'm planning to set up a lab in Jodhpur, Rajasthan for mushroom spawn production. I tried oyster mushroom at pilot scale, got succeeded to culture, but market is very uncertain to how package fresh mushroom and sell directly in the market.
So the consultancy of ours is free for that. You can directly, communicate with our admin. His number is given in the group, so you can ask him. He will help you about these things. So you can call him later on.
The last question is what else can we use except wheat as a substrate?
So you can use different things as substrate. You can use millets. You can use corn. Yeah, there are corn cobs also used. Wheat is used. Brown rice is used. But a mushroom is widely used. I'm sorry. I mean, wheat is widely used. It's good, and it's mostly preferred. Now we have discussed why it is preferred. So you can use anything. You can use other things like millets and corn, but the chances of succeeding with spawn is higher with wheat, so go with it. So one more participant has asked ki what we can use for sterilization. What you have used for sterilization. So we have used 70% ethanol for it, and for any sterilization process, 70% ethanol is only used. So you can use 70% ethanol for sterilization. Uh, this is the QR code. You can apply for your certificate via this, and all the students who are going to apply for the certificate, you will be provided with this lecture and all the notes via your email. So please, if you find this valuable and if you want go on with spawn production, so sometimes you need a certificate because there are so many programs run by government of India, subsidy programs, and for that students need certification. So you can go on with I mean, if you find it helpful, you can take the certificate. Yeah, I think there is one more question. Let's take the question.
How long can we store substrate? What is the optimum condition to store them?
Substrate. I mean, you want to know how long can we use wheat? Yeah, I mean, if you have boiled the wheat, of course you cannot store it, because if you will store it for longer period of time, maybe if you're storing for 3 days or 5 days, and if you cannot maintain that moisture content of it, of course it is going to reduce your spawn mycelium productivity, because moisture is a really important factor in that. So you cannot, if you could maintain 50 to 55% of it, you can store it, but it is really difficult to maintain that. So basically it is preferred that once you have boiled the wheat, next you are going to treat it with the chemicals, and after that you start the spawning. As we have discussed, either mushroom production or spawn production is not for people who want to delay it for tomorrow and tomorrow and tomorrow. It's not going to be a very tedious and hard working thing, but it's going to something that you need to schedule your time and you need to do it every day. Next question. Okay, there's one more question popped up.
What will be the costing to set up lab for a spawn lab?
If you want to set up a spawn lab, I think I won't be able to describe all of these things here. So you can call our admin for that. If you need consultancy, you can call. We provide consultancy for that, and it's free of cost. So we are not going to charge anything from you. For you can call anywhere after 2 p.m. to in between 5 p.m. You can call him, and he will be dealing with your these problems. So don't worry. I hope there is no further question.
Another question is, how can I know that the wheat has reached 50 to 55% moisture content?
So basically, there is equipment called moisture meter that is used for checking the moisture. But over the period of time, if you have started growing spawn, you will touch and you will know that yeah, it has right quantity of water. There is one more question.
Another participant is asking, if it's completely free, the workshop, then why is there 299 rupees for e-certificate?
So you think we are operating free of cost? No, we are not, of course. No one can operate free of cost. So there are certain amount of charges that we also have to pay for, the certification, the processing charges. We also have to pay, since we are conducting a workshop, we'll have to pay a certain amount of charges to government for that, because you cannot do all these things free of cost. Of course, you will have to pay those charges to the government, and people who want to join it only for the educational purposes, it is absolutely available to them. And people who want to take certificate, I don't think rupees 299 is very much for them to pay and to take the certificate, you know and it's for 3 days. 3 days and 299, I don't think it's a much price, and I think it's worth. You can pay. There's one more question that has been put up.
If we dry the grains after boiling, the post-boiling grains treatment with chemical is not needed, sir?
Calcium carbonate and calcium sulfate is used as a supplement, so we need supplement. As we have said, we need certain kind of supplements. We need to maintain the pH, and we need to provide sulfur and calcium as supplement for it. So you need that. You need to maintain the pH. That's very important. There's one more question put up.
So while selecting alternative substrate, what should be considered as major example we need to maintain the condition?
I think there is a question posted by Dr. Lata. Please post your question in the group, because I am unable to understand what you're trying to ask. Please post that in group. We'll try to answer that. I'm finding the question to be incomplete. There's, I think, one more question.
Another
There's a
question
question.
is asked that Gucci mushroom can be grown in India in laboratory conditions.
Any kind of mushroom can be grown in laboratory conditions. It's just that you have enough of equipments and support that you can provide all the environmental setup that is needed for that. So if you could maintain the temperature and pH and right kind of moisture, you can grow any kind of mushroom.
And next question is from Fariha. She is asking that they sterilize the tub in which the filtering of wheat was done. What is that they used for sterilizing?
We use 70% ethanol. So for any kind of sterilization process, 70% ethanol is used. You can use 70% ethanol.
We have another question from Varsha. She is asking that what are the advantages of mushroom spawn cultivation?
So first and the most important advantage is that the gap in market in mushroom and spawn cultivation is really high. So one thing that absolutely supports this. The next thing is that for spawn, mushroom cultivation, you need real man. It's a tedious thing, so you need manpower. For spawn cultivation, that much of manpower is not needed. It's not that laborious, and you can set up that spawn production plant in small space. For mushroom, you need a bigger space, more sterilized environment, and you need to set up AC and all that. For spawn, it is not that necessary. So, and it requires more profit in the same kind of investment, small investment and more profit in comparison to mushroom cultivation.
Another question is where do I get inoculum for the substrate?
So you can get inoculum for the substrate from us, or from any kind of I mean, you can search other markets as well, but you can take that from us because we provide spawn. So one thing that you need to make sure that from wherever you are taking the spawn, the source need to be trusted, because as we discussed, your quality of spawn is going to decide the quality of your mushroom. And most of the time, why we said that there is a huge gap in mushroom cultivation and spawn production, because the spawn that is available in the market is not of really good quality. So if you could provide really good quality spawn, uh, automatically it is going to boost the production of mushrooms. So that good quality spawn is really needed in the market. So buy from anywhere where you can trust. We also provide spawn, and it's a trusted place. You can even consider us.
Another question is why do we choose spawn cultivation over the mushroom cultivation?
Because in the small place you can start this. It needs lower kind of investment in place of mushroom cultivation. Mushroom gets spoiled in, once it is harvested, within 4 to 7 days it start getting rotted, and after that it has no value. Even some people say that you can dry the mushroom and sell in the market, but it's not that really profitable. So it even receives less. It's really difficult after drying. Becomes really difficult to even retrieve the amount that you have invested. Eh, but in spawn, if you have significant amount of market, it does not get spoiled easily if you could store it properly. So in small, you can start it in small place. Its shelf life is higher. Its market has a huge gap in the market. So it's current time it is preferred over mushroom cultivation. So that is why. Otherwise, mushroom cultivation is also fine. So I hope, I mean, if you have any further question, you can ask, or mm, anyone, if you want to ask something. So if you don't want to, I mean, if your questions are over, so we'll start tomorrow's session at the same time, 5 p.m. And tomorrow's session is even going to be more important, because tomorrow we will do we'll mix the chemicals with the dried substrate. After that, we'll learn how the process of bagging is done, how it is packed properly. So it has aeration, but I mean it has good quantity of aeration also. And after that, how we sterilize it. So tomorrow's session is going to even more important, because tomorrow we'll see how spawning, before inoculation, all the process can be seen tomorrow. So it's going to be a really interactive and important session for all of you. So I hope it was a nice session, you would have learned something. So let's join tomorrow and keep learning. Thank you so much. And if you want to take the certificate, you can scan the QR from here and take your certificate, register for it. Thank you so much. So we'll be sharing a feedback form in the group after this session. So you can fill the feedback form and you can let us know that what else we can introduce into the session for tomorrow's session, apart from our lecture and live demonstration, something that you want to know about. So please do that. We love to know about your ideas and your comments about this. So thank you so much.